And welcome from my laundry room. Thank you all who visited this after show for the debate. I did invite Carissa to come, and also Dan Stern Cardinal said if it's not too late, he'll join in, but I didn't expect the debate to go 3 hours. It's already 11 o'clock. So Dr. Dan, if you're listening, I did send you an email, same with Carissa. Otherwise, I'll have to do this solo. I don't, obviously, I've been extremely busy and trying to get ready for a delayed enrollment PhD program in biomolecular engineering. I gave a little bit of intro about some of my work. I used to be in the defense contractor before becoming a biologist and a student of physics. And so now I'll be studying biomolecular engineering. It was great to see Jen. She was actually she was my tag team partner in a debate against evolution back in December, and she's on the other side. She just has a beautiful personality. Unfortunately, I don't agree with kind of her scientific view of things. I was grateful we had an opponent. I couldn't have found a better buffoon who thinks so well of his knowledge about evolutionary theory, and he doesn't even understand basic molecular biology. It was kind of fun to debate that. A little frustrating, but I tried not to let it get to me. I'm sorry about that. You didn't mention your channel at the end, so it was hard to find. Yeah, I did make that mistake. Thank you for coming here. So this channel is Evidence and Reasons for the Christian Faith. I thought Carissa really, wow. She opened with something really personal. She talked about her boyfriend dying of an overdose. She did talk about that on this channel earlier, and that's just kind of heart-wrenching. And she's talking about how her boyfriend was into Darwinism. It led to him having really dark thoughts, and it fueled his drug addiction or his substance abuse issues, and it led to his death. She related, at least on this channel, she alluded to it a little bit in the opening statement, how she was digging through her boyfriend's belongings and finding all this stuff that was very dark. And it was steeped in Darwinism. Darwinism as a science doesn't offer a lot of hope. I mean, even though science is neutral, we can see certain theories that actually alleviate human suffering. And I showed the 5 laws of nature. As we've had control and command and understanding of these 5 laws, which include electromagnetic theory, life has gotten better. I can't say the same for Darwinian evolution. It's not brought any benefit to my knowledge. And that was echoed at her very personal level, that she saw it ruin the life of someone she loved. And so this was quite personal. And this happened before she became a Christian even, or a creationist. So I was really proud of her to come forward and say that, even though this wasn't about science. I think there were a lot of people in the audience that probably could relate, that would be appreciative of someone, just pouring out their soul and saying, hey, I believe in evolution. I wasn't necessarily a Christian or creationist. This is where Darwinian evolution, where it led to someone's death. And that's not totally uncommon. I mean, maybe it's rare, but it happens. And she was relating how that motivated her just to study the question further. I'm very impressed that she had worked for the California Academy of Sciences, and she had National Science Foundation grants to help her. I haven't seen Dr. Dan come up yet. I don't see him in the studio. So Dr. Dan, maybe this was too late for you. We can definitely continue the conversation. There were a lot of things, given the format of the debate, where we couldn't really explore things in depth. This whole thing about protein evolution is a very serious topic, and my opponent was just dismissing it. Oh my goodness, Dr. Dan.
Hey,
honored. Look,
what's up?
You've told me the show because I promised everyone you'd be here, and I know it's so on a Friday night.
No,
And
it's a little late, but I figured I'd pop in. At the very least, I'd say hi, hear about the debate from you. Is Carissa going to be here? Because I'd love to chat with her
totally
too.
invited her. She had been worn out. She was doing tours at her university today for new students, and you know how
That's
might
busy.
be.
Yeah, I'll do it.
So if she doesn't join us, maybe sometime we all could talk.
I would love to. Yeah.
She definitely explored the young Earth creationism more than I'm comfortable with, and people know that I look at young Earth creationism, but I'll probably side with you a lot. Your gang, and say, hey, I think some of your criticisms are pretty pointed, and I don't have good answers for them. She's a little bit more on the needle on her meter. Is that, I mean, both, just for clarity, both of us believe it, but I don't think the evidence is quite as strong as she might think.
Right.
But it was still, it's great to hear her talk about it, and I do know that she's doing research about that. But that being said, I just want to say how much I appreciate you, Dr. Dan. I know, I mean, I hate being so disagreeable to something that is something you've studied and devoted your life to, and I'm just trashing it there, and I feel bad about that because I love you to death.
No, I love, there was one point where you were like, and Dr. Dan, thank you, I appreciate you, and then in the next sentence you were like, and also evolutionary scientists are liars. It's like, thanks. And that was funny. Just the juxtaposition was funny.
Okay, I look at them like Richard Sternberg. I never said they were liars. I think they believed in something that's wrong, and that's not quite.
So I'm delusional, is what it is. But I'm a really nice guy.
Well, I'm mistaken.
I'm mistaken.
Okay. Because I make math mistakes all the time, and I remember something I was working on for a long time. It's like, isn't this working? So I just wanted to say how much I appreciate you coming here and inviting me to your channel. And
thank you for the invite. I appreciate it.
You have given such very thoughtful criticisms, and you're one of the few people that have gone through great lengths to characterize my position accurately. I was really impressed with your last show about me, even though I wasn't.
I tried.
We had one show where I was on your channel, and then the subsequent show, it was just a monologue. I was really impressed.
Thank you. Yeah, I mean, I try, right? Because what's the point? So this is actually a really good topic, actually, because this is what I was hoping to talk to Carissa about, because, like, you're right. What I'm always trying to do is, like, and not just when I'm talking about you specifically, like, anytime I'm trying to address something that creationists are saying, I always start with, like, here's the argument, here's a source for this argument, here's where you can go and find someone making this argument, right? And that's kind of like how I frame everything. It's like, here it is, now let's talk about it. And, because what's the point of, like, arguing against something that is not representative of what the opposing party is saying? Because then that opens the door to them saying, that's not my argument, and then everything you just did doesn't count, right? Like, so what's the point? So you have to be accurate, just, like, putting aside just, like, out of decency, accurate representing your opponent. It doesn't make sense in terms of communicating the arguments to do that, because you don't accomplish anything if you're not accurately representing the thing you are arguing against, right? Does that make sense?
Oh, yes. I just want to express my appreciation that you've gone like the extra 10 miles to make sure you represent me accurately. Sometimes in the process, you catch something that I didn't say, that I didn't formulate very well, or even that it was just outrightly wrong. And that's led to retractions on my part, and I'm extremely grateful for that. Just so you know, part of the reason I'm doing that, Carissa and I are thinking about writing an intelligent design or curriculum for college students. She being a college professor, that's something on her heart. She wants to go into young Earth creationism. I was like, well, I'd probably say, okay, here's the good and the bad about that. That would be a balanced course. And I felt it was very important to hear criticisms from, evolutionary biologists like yourself if we say something inaccurate. And even if I formulate my own arguments poorly and you've called that out, I'm just like, well, it hurts to find out you're wrong, but it's better I catch it. And you've done an excellent job of that. When I stumble on my words, you've asked pointed questions, and I'm just like, I could tell I was fibbling, I'm stuttering and not formulating my words. I said, I haven't probably thought that through quite as carefully. And you posed the question that really stumped me. And I'm very, very grateful for that. And these are very honest questions, by the way. I just wanted to say, this whole thing about, oh, you didn't give me the link. I'm just like, dude, I'm so busy in the middle of the debate. You can rewatch it. You could see the citations, and he's accusing me of dishonesty. You don't do that to me. And I just wanted to say how appreciative I am.
I try.
Yeah.
I try.
Okay. And I do have one more thing to say that I think you should know. I cut you off. How about I hold my thought and I'll let you talk?
No. Go ahead. Finish your thought. All good.
So you've been critical of John Sanford and some of the things he said. And there are times when I say, John, there are ways you phrase things and you're going to be walked all over. And I don't like seeing it. But, so one of them was about like this whole thing about viruses. He and Rob Carter.
Yeah, the H1N1, the famous H1N1 paper.
Yeah. So, before he gave his talk at the NIH, I said, they're going to pounce on you on that. And, I don't think, my role was to tell him, where I think he would be vulnerable. And so I've appreciated, you've said very kind things about me, and yet you said some really horrible things about my boss. And I wanted to say, hey, John, it's a little different when you're talking to a very hostile audience. You have to watch your words and formulate your arguments with wording that probably would help you a little bit. And there were some things that with what I call genetic entropy 2.0, which was a little bit released in our Springer Nature reference chapter, that have changed since he wrote the book in 2004. But I just have noticed how much more nicely you treat what I say versus my boss. And I just want to say thanks.
Thank you. That's fair. I am definitely very harsh on John Sanford. And the reason for that is not to put too fine a point on it. I think he's lying a lot, specifically about like what Kimura said and why he said it the way he said it. Because like he'll represent Kimura one way in the famous 1979 paper with the distribution of fitness effects. And his explanation for why he does that is contrary to what Kimura writes in that actual paper. And I find that indefensible. And that's why I'm very harsh with regard to John Sanford, because I think like he is being dishonest about the things he is representing. And that I take offense at that. I think that is inappropriate.
Well, I'm appreciative that you're leveling with me, and this is incredible that we're having this live public discussion and everyone's hearing what we're actually saying. I will say this, having worked for him, and I mean, for him, we still are colleagues. He's getting up there in years, and that's affecting things. And I'll just leave it at that. He's never said, "Hey, Sal, we know this is false, but let's push it." He's never done that to me. I could just say that. There are times I said, John, I think this is wrong. I told him his stuff about thermodynamics. I said, I think you got bad advice. I think it's wrong. They're going to pounce on you for that.
I'm not expecting or asking you to defend anything. I just
wanted to
from there. Yeah.
No, I wanted to also, because people would be curious. I do want to acknowledge someone here. I was in Pennsylvania recently, and I was at a creation conference, which I rarely attend. And she was someone I met at the creation conference. I can't believe she found me at my channel. And here I am having a discussion with a real live, honest to Darwin evolutionary biologist.
Well, okay, it's great you said it that way, because here's my big takeaway from what I caught of the debate. I was popping in and out doing other stuff, but I caught chunks. And every time I listened, what I heard was lots of arguments against Darwinism. And it was mostly from Carissa, not you, to be fair. So I don't want to, ask you to defend an argument you did not make, right? I don't want to do that. but like you know that modern evolutionary theory is not Darwinism. Like I know you know that because we've talked about this. Like I know you know that. So when someone says like Darwinism can't explain X, there's no Darwinian mechanism for Y, right? Like you, like Sal, like you know that's not on the level because we've talked about this, and you know like how our understanding of mechanisms of evolution have changed over the century and a half since Darwin was doing his thing.
And,
Like,
well, I was specifically talking about the when I invoke Darwinism or natural selection, and I, correct me if I'm being unfair here. There are times you invoke natural selection too.
oh yeah, natural selection, totally fair.
And so
that's totally fair.
And so, as far as we're, okay, so being an engineer, what I don't know if you've read Darwin's Dangerous Idea by Dennett. He has chapter 8, biology is engineering. That actually resonated with me, even though it was very pro-Darwinian and anti-ID. It was actually it was pro-design, but not intelligent design.
But like Dennett's a philosopher. He's not a biologist, right? He's not a practicing biologist. And if you ask, me, or like, for example, my friend, Dr. Zach Hancock, right? Practicing evolutionary biologist, or any of my colleagues that I work with, my PhD advisor, right? Like you look at people who are doing evolutionary biology and you ask them about like Darwinian evolution, they're going to tell you like variation with an indeterminate cause, because Darwin didn't know about genetics or anything, and natural selection. And that's it, right? Like there's stuff after that. There's neo-Darwinism, the modern synthesis, which is people, because of the name, the modern synthesis, think that's representative of modern evolutionary theory, but it's not. That's from the 1940s. And you've got a whole bunch of stuff since then. If you ask me personally, most importantly, things like neutral theory and nearly neutral theory. And if you're saying Darwinism can't explain biodiversity, right? Or Darwinism can't explain increases in complexity, then you're leaving out a whole bunch of mechanisms of evolutionary change. And yeah, natural selection, like if, this is like a gotcha in creation circles. And I know you know this because you know all the arguments inside out, right? You'll hear this all the time. Natural selection doesn't create anything new, right? Natural selection just removes variants that are suboptimal, that are less fit. That's totally true. But that's not the entirety of the mechanisms of evolution. So to say like Darwinism can't generate new complexity, Darwinism can't, do X, Y, or Z, like that doesn't make any sense as an argument against evolutionary theory writ large.
And by the way, I appreciate you pointing that out, because there was something I left by omission just by now that you mentioned neutral theory. In the course of a time debate like that, where, oh my goodness, you had 1 minute, I'm just like, you're kidding.
Yeah, it was short. There were short intervals. Yeah.
I was like, I do not have the opportunity to kind of be a little bit more, cross the I's and dot the T's. I mean,
yeah, I'm mostly not talking about you here. I'm mostly talking about Carissa, and she's not here to defend herself, so I don't want to like go too hard on this. But like, you could say evolutionary theory can't explain X. You can say like modern right? You could, but like presenting evolutionary theory as Darwinism is just a misrepresentation of what evolutionary theory is, and leaves out a whole bunch of things. And like the repeated invocation of I think at one point there was the actual wording that right, successive slight modifications, right, is the wording if you go back to 1859, right? And like, yes, there are systems that you would look at a trait and say you couldn't do or wouldn't expect to see A to B to C to D to right, successive slight modifications to get to that thing. There's other evolutionary pathways to that thing that are non-Darwinian. So saying Darwinism can't explain it is not an anti-evolution argument. It's a description of which mechanisms apply in this or that situation, right?
Okay, so there's a productive way for even Carissa and I to benefit from what you just said. And, because she's a university professor, I expect she would want to teach evolutionary biology as accurately as possible, especially if we make a pro ID
I hope
course.
so.
Yes.
I would hope so.
So let me just throw this out there for you, Dr. Dan. If we start to write chapters of this, you're welcome to invite you and your colleagues to just take a look and say, hey, you totally misrepresented this. I personally will go back and say, okay, well, what would you suggest to represent this more accurately? So going back to the debate, I really wish I had time to mention neutral theory and near neutral theory, because I have studied it, and actually it's been an argument against there was a paper by Jukes and King called Non-Darwinian Evolution. It's kind of a form of and so just to complete the circle, I actually don't think neutral theory can explain the emergence of complex systems like what Behe is talking about. Certainly not the systems that I've studied. So it's a little bit, there's not a lot of literature on why neutral theory would not be able to explain that. I accept that it is a way that things evolve and it has been observed. And maybe that's something that could be worked on my part. And I'm grateful you pointed that out. And so, a future anti-evolutionist textbook that probably have to be something there. So I do know of the third way and all this stuff with.
I know.
It's like, well, it's kind of hard to say it's a unified theory.
It's the third way, though, in the actual field of evolution. Like, there's a bunch of cranks that it's like the third way people and the evo psych people, and none of no real evolutionary biologists think a lot of the third way people are more like philosophy of science types that don't have a background in actual evolution. And I don't want to just say oh, they're not in the field, they don't they don't count. Their arguments, they're saying, for example, evolutionary theory doesn't take into account X, Y, and Z. But you can go back to papers from the 80s that are dealing with the things they're saying we're not talking about. It's just, it doesn't make sense to me how you've got these, the kind of these people saying, and again, largely from a philosophy of science background, saying evolutionary theory doesn't deal with all these different things and we need to rethink evolution. And man, I don't know. I'm immersed in this field and people talk about that stuff all the time. It just doesn't I just don't know how to deal with that. And that comes a lot of creationist stuff is like that too. Oh, you can't explain X, Y, Z. It's here's 12 papers on that exact thing, right? Here's a bunch of stuff on that. I'll use an example that you're familiar with, right? You referenced at one point the genome, I forget the exact name, but the paper is something like genome reduction is the dominant mode of evolution. I forget the exact name of the paper, but I think you know the one. Yeah, you know the one I'm talking about, right?
Wolf and Kunin.
Yeah, and it's a famous paper and it's an important paper. But creationists bring this up, you bring this up, and I don't get it, because what that's kind of talking about is how you have duplication events of various scales. And then once you have duplication events, you've basically increased the size of the genome to the point where the energetic cost of maintaining it becomes a problem. So now the parts that are excessive that you don't need, when you experience deletions of those parts, there's no cost to it, right? So now you end up, you expand the genome and then cut away the parts you don't need. And what's left are the parts that when you duplicate them, provide some benefit. Either you have an extra copy of a gene, you have more of an enzyme, you do a thing faster, or those duplicated parts mutate and now that enzyme can have a different substrate, right? So you have things like constructive neutral evolution where you have a process like duplication that is just as a baseline, it's neutral, right? You can't say anything about the fitness cost without seeing what actually happens, right? And then you're going to have processes from there that are going to be on net reductive, but the net effect of all of these processes together is to increase genomic complexity and to increase the net functionality of the genome. And that doesn't involve Darwinian evolution, except for the selection part, in that one little bit of it. All the rest of that stuff is non-Darwinian, right? It's neutral or nearly neutral. And that's an increase in complexity and an increase in function.
And well, see, I think the fairest thing, by the way, I wish I could, I'm glad this is being recorded because I think, like even on my academic channel, I wanted you to have the chance to just, that's why I didn't do like one of these things where I'm playing back your video and then I'm just
Right,
interjecting
right.
comments. I want people to hear you have the platform fully, and then I'll give my rebuttal fully. I'm not going to
Yeah,
just
yeah, yeah.
chop. And I wanted to honor your work. If I were making a college course, I would want to have your viewpoint, what you just articulated, in my view of that. Now, the way I viewed that, you kind of got that when I put my little comment there. I said, these punctuated changes I said are unexplained. What I mean by unexplained is we can come up with a theory, but one thing that, in that I'm seeing is there's huge metabolic costs for very complex organisms, and one believing genetic entropy. I think we're going to see a lot of complex species go extinct. It's just, it's the whole thing about the peacock's tail. There are a lot of architectures that are just metabolically, the reproductive cycle is very long, and I just don't think that anything, actually natural selection will work against it. That's the one time I say natural selection is actually pretty active. It's going to be ripping apart complex systems. So I'm just articulating, this is my thought. We don't have to argue about it here. I think the way to do it in university is I would love to let you to have your opinion hosted, like say on a website, YouTube channel, and say, my editor's pick, Dr. Dan will be the go-to guy for a lot of this, and then you could recommend people, and then I'll give my arguments and just let the students hear.
I mean, I will tell you that, like, first of all, I'd be flattered that you think I'd be a good person to do that. But also I don't think, if you want, like, if you have like a book that's geared towards intelligent design, you're like, here, edit this. Make sure the evolution checks out. Everything's kind of reasonable in terms of, kind of up to date with evolutionary theory, accurately represents it. You look at all the intelligent design stuff that's out there, right? Mostly from Discovery Institute. They're the ID people. I mean, they do a terrible job representing evolutionary theory. They're Darwinism this and Darwinism that. It's all the same thing. And Michael Behe's told me to my face that, like, oh, well, I just call it Darwinism if it's undirected. That's
Okay.
That's dishonest. That's not what that is, man.
Oh, all right.
Okay. So
I
feel like there wouldn't be a lot of the chapter left after I was like, going through it with a red pen, right? That's the.
Okay. It hurts me to hear you say that they're dishonest, but you know what? I can't control what they say.
No, and I'm not asking you to defend them.
I'm not going to try to defend it because there's a nice solution. I'll just say, look, Dr. Dan, what would be not offensive to say? And I'll say, I'll try my best to do that. So at least as far as you and I go, I can try to use the best language I can. And then we may disagree about the usage of words, and we could just note that. But I'm going to go the extra mile and say, look, it doesn't hurt. It doesn't help our case to be accused of stuff like that. This is easily fixed.
I agree, and I would like to see creationists do more to kind of keep their own house in order, if you will, because there is just a lot of, misrepresentation of fairly basic things that you could just not misrepresent, and it wouldn't have like big implications. And like the Darwinism thing is one of them, which is like a branding thing. And Nathaniel Jeanson has been like upfront about this. Like, why did I call it replacing Darwin? Because it's snappy. I know Darwin's not, modern theory, but like you got to get people in the door, right? And this is particularly an issue with intelligent design, more so than the more overt young earth ministries. But like, it really is about marketing and like intelligent design. My opinion of ID. Like, my opinion of ID is that it is a marketing campaign first and foremost, and a scientific thing after that. And like, it's frustrating. It is frustrating to have people who are saying this is a scientific conversation, who are not accurately representing the content of the science in question. I find that very frustrating.
Well, okay. All I'll say is I'm not going to defend their, I wish. There are things I've been critical of the young earth community, and I get in trouble from them, and frankly, I don't care, especially if it deals with Hovind. I can't believe someone was comparing me to Hovind. I was so offended. That's neither here nor there.
No comment on Hovind.
There's some things that are easy. I think there's some things that can be fixed. We have conversations like this, and we can do our best to fix it. That being said, there is a focus. Just as a FYI, and we'll move on to other topics, because I did promise we could talk about some other things that are not even in the debate.
No, I'm happy to. I actually have. There's two things I want to bring up related to this. That after you, so please go ahead, and I'm happy to.
So we still have the issue with Lenski's Avita program and the Weasel program. The computer scientists are very interested.
Mm-hmm.
We think this Avita program that's building complexity, it was falsified by some of Lenski's experiments. It's like, well, even if we're not saying it's a general trend that genomes decay despite sustained fitness gains, it's like, well, how does Avita account for that? How can you characterize situations where you're going to have gene loss by natural selection? Because we have now we're starting to see papers that talk about gene loss is adaptive. And I'm just like, well, long time no see. Maybe we don't have a handle on how extensive that is. I would argue, and I'm just postulating, conjecturing, it's actually going to be large scale. We'll find out in 40 years if we keep sequencing. We are interested in, basically false. So you're going to see more anti-Darwinian stuff coming from. I'm actually working on it because they're likening it to genetic algorithms. I'm not saying that that's the only. I'm agreeing with you that that's not the only mechanism of how evolution proceeds, but it will still be a hot topic because Lenski's Avita program really has still gotten under the skin of the computer scientists and the ID community. I'm involved in one paper on that. So I just wanted to pass that on to you. We will keep hitting up on that.
Okay, I have a question about that, but before I get to that specifically, that kind of work there. So like talking about making good arguments, making honest arguments, making sure that the argument you're making accurately represents the other side. Here's where I'm gonna come at you a little bit.
Okay.
Because there were two things during the debate. I'm like, I feel like Sal knows better on those. So one of them, which you keep bringing up, is the if encode is right, evolution is wrong thing. Which, so you know the famous Dan Grauer talk at the conference in 2013. If encode is right, evolution is wrong. He goes through, gives a great talk. I know he gave a great talk because I was at that talk. I was in the room for that. I was there. It was one of the best talks I've ever seen at a conference. Now, he wrote a paper on that in 2017, talking about the maximum functional fraction. And I know you've cited that paper because you talk about like the bonkers equation, right? And how many kids somebody would need to have given the math in that paper. But there was a follow-up paper in 2020 on that, where the authors found an error in his math and corrected it. And Grauer was a reviewer on that follow-up paper, agreed, published without revision, said, yep, your math is right. I was wrong. Checks out. Now, if you go to the 2017 paper, there's a correction at the top saying, hey, this math is not right. Here's the updated paper. This is the correct math. Right. And now the whole like if encode is right, evolution is wrong thing, that's not what he meant. I know that's not what he meant because I was in the room, right? The way it's presented by creationists is not the argument that he made. And I know that because I was there. But even assuming it is, that math no longer holds because of the 2020 paper. It's a Galeota-Sprung 2020 functional fraction human genome something. And like now it's 2024 and you and other people keep bringing that up. Even assuming that the framing is correct, the argument is no longer valid because the math that it rests on was shown to be wrong and has been corrected. Like, so
I do have a response to that.
You should stop saying that.
Well, no, I have to disagree here, Dr. Dan. But maybe okay. Let me just say, even if you disagree with me, I want to show that I had honest intent.
Okay.
Because this and it ties to this whole discussion about what fitness means, because
No, it doesn't.
no, I wanted to point out, because I had derived this. It was in our 2021 Springer reference chapter where we actually cited, and I worked through the math from Kimura, all the way back from Kimura. You just take the Poisson distribution, which Mueller used. It's that simple. If you're defining a defect in the functionality, those numbers actually hold. That's where you get, and it's backed up by the papers. When you look at the equations that I put there, where you take the Poisson distribution, it agrees with Nachman and Kroll. When you have 3 mutations, you need 40 females. It exactly agrees with the formula I put there. And then also Walker-Keatley. And then just extrapolating it out, it came out to the one that Dan Grauer came out. So
did you?
This is a simple of math derivation, and I've done the derivation right on my channel.
But it's wrong. The math is wrong. Did you cite the 2020 paper in the book chapter?
I don't have to. The Poisson distribution is accepted all the way back to Kimura.
Yeah, but you didn't cite
Poisson. I don't know why that's in dispute.
You didn't cite the revised math that everybody agrees is correct. You didn't cite that in the chapter, and I know you didn't cite it because I read the chapter and reviewed the chapter, and I checked, and it wasn't there.
Okay,
you didn't cite the updated paper even to say you didn't cite the updated paper even to say that it was wrong, and here's why.
Okay, so I tell you what, I mean, the fair way to settle this, okay, is that I will look, you sent that to me, and I have to tell you, I didn't read it because I was quite confident my calculations were correct. Okay, maybe that's arrogant on my part, but I'm just like, this is a Poisson distribution. This is so fundamental to our understanding. If you want to undo all of the evolutionary biology that relies on the Poisson distribution, because I did the derivation on my channel. So in fairness to settle this, I will, if you can kindly send that. I think it's only right that I review it and read it, and because I value your criticism, and if you want to come after me on that, I think that's fair.
To be fair, I have brought this paper up to you before, including the second time we talked about genetic entropy on MDD way back in the day.
Yeah, I'm confessing I totally disregarded it because I had this is how little I have regard for Dan Grauer, anything he said.
Yeah, but the thing is, the whole point, see, you should have liked this paper then, because the whole point is that Grauer was wrong. That's the whole point of the paper, is that Grauer was wrong. So the fact that you don't like Grauer should make you like this paper. It showed that he was wrong. You should like that one.
Okay, so
so there's
so I'm acknowledging you sent it to me twice. I confess I totally disregarded it because my opinion of him is so low. I just don't want to waste any time reading it. So and I'm confessing that I should have given more, and especially now that you're bringing it up, I'm not going to make an issue of it, and I'll say I'll put it on my things of list to do, and it's only the right thing to do. I have to set an example for the university students if I'm writing. And so that's just the right thing to do. Okay. So that's why I welcome your criticism. And if I come to the conclusion he's still an idiot, which I think he is.
Well, I mean, that's fine because he's saying
I will have done my due diligence.
The whole point of the paper is that his math is wrong. So I don't think that's not the thing that would change your mind. The problem is that it was directionally wrong in the opposite way that you want. He was wrong in such a way that he was harsh, right, for evolution, and the numbers actually are easier, right? But the point of the paper is that Grauer was wrong. And he acknowledges that. He says straight up, like, yeah, this math is right, my math was wrong, and there's a correction on the original paper.
So I will look at the correction and I do apologize to you, and I hope you'll, so I mean, you had the right to call me out on that, and I'm and I
hope you don't take it personally. This is not,
No, and I'm trying to explain to my audience. I'm saying Dr. Dan is right to call me out on it, and it's not because I was trying to be dishonest. I just have such a low opinion of Dr. Grauer, especially the way I don't need to go there. I have a very low opinion of him. And that opinion keeps going lower the more that I read his stuff. So, but that being said, if I have to set an example and say, look, just because you don't like someone or value their opinion, if other people do, I have to study it. So that settles it. And, I will look at it. Thank you.
Okay. I got one more thing I got to bust you on here. All right. It's the protein orchards thing. When you like to cite me as saying that proteins do not all share common ancestry, which, as we've talked about, we agree on. And here's another soundbite for you. Proteins, hang on, let me point at the camera. Proteins do not all share common ancestry. You can use that. But here's the thing, Sal, why does every evolutionary biologist say that proteins don't all share common ancestry? Because you're not going to find one single evolutionary biologist that says they do. We all agree on that.
Oh, and this is the thing that
why not?
This is why I'm pointing it out is let me just share this. When I told Carissa that, she said, "Don't you?" I'm amazed that he doesn't see that there's a problem. That's what from our side.
I'm amazed you don't see there's a problem.
Look, I work with protein biologists.
Mm-hmm.
And I shared your clip with some of the people that
you've
seen me on. And frankly, some of them just laugh that you don't see a problem.
Yeah. Well, okay. Share this one with them. Why do proteins not all share common ancestry?
When you put, I will give you my answer. When you put it,
let me hang on. No, let me rephrase the question. Why do all evolutionary biologists agree that proteins don't all share common ancestry?
Because the bioinformation, there's no homology.
No, that's not why.
Okay, correct me on that. Because
it's because de novo protein coding genes are a thing, and we've observed them evolving.
And now
that's why.
I don't want to.
There's a real thing that happens that we've directly observed.
I don't want to. Okay, I'm glad you're coming after me on this one. We're going to have a good conversation now.
Let's do it.
I have a contact in the NIH, and I'm not going to out him. And we both think that he will come out of the closet eventually. Okay, just wanted to say. So it's. I'm not arguing this in a total vacuum.
That's
but
fine.
The citations, and I read. Okay, I didn't. You gave a list of citations, and I didn't. In your talking about the protein orchard. By the way, I was really impressed. I just have to say this. You said Sal had we had the created kinds, and that's an orchard, and Sal borrowed the language to describe proteins and used the word. I used the word orchard. That's a creationist thing. I kind of like the metaphor of orchard, but I want to credit you. You're the only one that realized that.
Oh, thank you. I think other people caught it too. I don't think I'm the only one that realized it. I just happened to make a video on it and say it. I think other people caught that, to be fair.
So some of the stuff I saw on de novo protein stuff, it's not necessarily the list that you gave, but I've obviously Googled de novo proteins, and it's very hard not to totally dismiss the papers that I see so far. And just like, I don't want to waste my time. There's Carvunis. Her name's Carvunis. It's a long name. She wrote a paper in PLOS on de novo proteins, and she cited Ono. And Dr. Sanford and I and Joe DeWeese have refuted that paper. And not only that, in 1992.
Oh, yeah, that was the frame shift paper.
Yeah,
right?
And it was just very easy to write her off at that point. I'm just like, okay, this is about the best they could do. But that being said, there was another paper I saw. It wasn't necessarily in your list. And the guys didn't even have confirmation that these are proteins. And then the other thing is, if you're only looking at RNA stuff and they're using ENCODE data, it's the same argument that these could be spurious. So and then there was lots of circular reasoning. I just got very tired of reading them, honestly, because it was the same thing on sketchy evidence. The other thing I just wanted to say is there's a false equivalent here, and it relates to irreducible complexity.
My favorite.
Okay, so there are where the weakness, I'm going to criticize my own side slightly here. Not all irreducible systems, I don't like the word irreducible complex. All or nothing is easier to understand, just like a password. When I've shared this with people and say, just look at a password, because you have passwords that are small, others that are very long. And you can't say that just because you solved the small password to say 2 letters, that it's going to be easy to solve one of 50,000. And so there's a false equivalence when you are able to make a translated mRNA transcript into a polypeptide and say that's de novo, and you don't know even its structure and function. I find that very problematic as putting that forward as something, as evidence that something as complex as a collagen, the collagen system, can emerge. So it's, again, so with irreducible complexity, and you're trying to get Scott Minnick to say, hey, that, you know, that system shows irreducible complexity can evolve. And I would say you have a valid criticism if they were all equal, if all irreducible complex systems were equal. And that's why I don't like using the word all or nothing, but with ranking. And so even granting that some of these papers were accurate transcripts, they don't solve the problem of the complexity level of, say, a collagen that has multiple post-translational modifications that have to be integrated to a system to work with the extracellular matrix. I just don't see it. And so I think we will cite the de novo thing. And my counter is that these are not equivalent. You can't, you know, the flaw in the ID argument is they're making all IC systems equal. And that would be as silly as making all password of the same level of complexity, just because you could then come up with a 2-letter password, call it irreducible complex, and say, hey, the whole thing about complex passwords doesn't work, you know, if they're like 10, 30, 40 characters long. And so, you know, that's one thing on our side that we have failed at, and you found that weakness, to your credit, and we're not doing a good job because we didn't frame our argument as well as it could have been.
I hear you, and I do want to, I would love to talk about that a little bit, but my point with the protein orchards thing is, if there are de novo proteins, which there are, we have documented their evolution, we can show that that's a thing that happens. You can even young Earth creationists will agree on it, because you can look at the distribution of orphan genes within what young Earth creationists would consider a created kind, right? And you could find places where everybody agrees they share common ancestry, but this one thing has this new gene that nobody else has, right? We can all agree that that is an event that happens. The level of complexity does not matter for whether or not the event, the de novo origin of new protein coding genes, happens, right? It can be a very simple, relatively short polypeptide. We might not even know its function. But if we can identify, hey, this is an open reading frame, here's what the protein is going to be, here's what the protein, the amino acid sequence is going to be, and we can isolate that protein from the cell, the fact that it gets translated to a polypeptide, you don't have to know the function to be able to say that thing is functional. And the reason for that is I think it was a paper from 2015, but I might have the year wrong, but it was a Michael Lynch paper, and the title of the paper is The Bioenergetic Cost of a Gene. And what they documented directly in this study is that the cost of having extra DNA in your genome that is neither beneficial nor harmful, right? A neutral bit of DNA, the energetic cost of having that DNA is below the selection threshold. So you can't select it out. The energetic cost of transcribing it is below the threshold. So if it's transcribed, you can't select it out. But if it's translated to a peptide, now the energy involved is prohibitive if it doesn't have another function, and you can select it out. So you can select against spurious translation, but not transcription. So we can document these sequences where you've got, you know, a bunch of rodents, for example, or a bunch of different species in the rice family, for example, that we all agree are related, and you can find protein coding sequences that are limited to just one of those lineages that, again, we all agree are related. The fact that they are transcribed and translated and maintained rather than selected out tells you that those are functional genes that arose since that one lineage where it exists diverged from all the other lineages, right? And again, this is something we know because the cost is such for the translation step in the process that if they were translated spuriously, that would actually cross the selection threshold, and you could select out the individuals that had a deletion of that region would take over the population, and that's not what we see. So we know from those types of studies that de novo protein coding genes are real. We can also see, like in organisms, that they directly evolve, right? We can document these events happening. And those two things alone, you can argue the point you made. I understand there's different levels of complexity. Certain levels of complexity are more accessible than others. I totally see your point on that. The length of the polypeptide, the number of interactions. I totally see your point on that. That is a different conversation from the one we're having here, which is you can't make a protein orchards argument, right? Oh, proteins don't share common ancestry. Yes, because de novo proteins evolve. And if we all agree on that, then it's not a counter argument. Like the protein orchards isn't an anti-evolution argument.
Okay, so I can modify the argument and say, no, I want to frame this correctly, because the proteins obviously that I was showing were very complex in all the interactions. And I'm saying you cannot, it is definitely, at least all the experimental evidence is like, you can't build this up gradually. There's so much stuff. I've looked, the collagen may look trivial, but when you look at all the necessary post-translational modifications and how it integrates into the system, my jaw just sort of drops. And you can't just, it's also unfair to my side to say, hey, we have evidence of de novo proteins. Well, it's like, well, okay, we're going to say collagen's de novo too. It had to be at some point, but does that make, does the set of modification, does that mean that modifications aren't statistical miracles? And I look at something like a collagen or, some of these transcription factors, I'm just like, or the insulin regulated system, even if you can build phylogenetic trees, even co-opting these parts, it just, from my side of the argument, this is a hard pill to swallow. So let me just say, I'm going to think on that. I'm going to say the protein orchard still, for the proteins that are extremely complex, it is a serious problem for evolution, because solving simple proteins doesn't solve complex any more than being able to solve a 2-letter password will solve a 50,000 character password. These are false equivalents. And this is the way I would articulate the argument. It's still, to me, it's unbelievable that something as complex as the spliceosome with all its proteins, it's like, oh, we found some de novo proteins here that solves these problems. The complexity is just too much. And I'm not the only one that's saying that. This was Michael Behe, who believes in common descent. He's like, yeah, this looks pretty bad. So I can modify it and say,
I hear you, but
I'm going to eliminate the simple proteins and say, okay, we'll all agree, creationists agree, de novo proteins arise, but is the de novo emergence for the complex ones?
So, okay, good. So that's a different argument now. It's not that you can't get new proteins. It's that
maybe I have to make it a different argument.
Right. It's a certain,
I'll have to identify it as a different argument.
So here's a question for you.
Would that be acceptable if I say this is a different, I didn't frame this as well as I could have. Dr. Dan pointed out that this was not really the
I would
best
love
way.
to see you make it a different argument. Yes, I think that would be preferable, because I think the argument as stated is not a good argument, because we've documented with solid evidence de novo proteins, right? Well, and that means orchards are okay.
Well, along those lines, practically every major protein family, even a creationist, say, well, that's de novo. We just might disagree on the mechanism.
On the mechanism, right? Well, yeah.
I'm going to
Well,
try to
here's a
reframe
question
the
for you, though.
argument so it's less objectionable. If it's a different argument, maybe what had
Yeah.
happened is the way I was articulating it made it sound like one argument when I was really making another, and that's a fair criticism.
Here's a question for you. Here's a question for you. Do you think collagen? Let's use collagen as an example, since you brought it up before. For people who don't know, collagen it's a triple helix. It's a fibrous kind of thing. So it's like a connective structural kind of protein. It's a triple helix of polypeptide. So it has 3 polypeptides wound around each other, and they have to interact, right? You have to have you have specific amino acids in one of those strands, right? Interact with amino acids in the other one that are biochemically compatible, right? So you have like a partial negative charge, partial positive charge. They're attracted to each other, and a bunch of those interactions stabilize that triple helix, so it works, right? Was that a fair representation? Like,
well, it is fair representation so far, but there are complexities with the post-translational modifications of the prolines. That is a big deal. Also, the glycolization, because it has to exit the cell, it goes through a lot. There are lots of signaling peptides all over it to put it in the right compartment to get processed. I mean, you can describe the final product, but it's not fair to try to say, well, that's all the complexity in it.
Yeah. Oh, no, that's not my point.
Include all of that. And then also when Robert Matheny was on my channel, he was pointing out, the extracellular matrix, there has to be communication through the cell membrane down to the nucleus, change the regulation and go back and forth.
Oh yeah, no, I know.
I'm just like, well, this is where my side is like someone like Behe, who believes in common descent. This is where we just start shaking our heads and just like, well.
Not my point though. My point is this: could collagen function with a different amino acid sequence? Could you have that function with a different sequence?
Yes, except for the proline, definitely except for the glycines, because the glycines every third. That's why it's a very noticeable pattern. It's very important for it, so it has to be there. There are other pieces there, so I wouldn't say it's 100.
No, I wouldn't expect it to be 100% variable, but it's not 100% locked either, right?
Right.
If you could have a situation where, for example, say you have trying to think, God, it's been so long since I took biochem. Say you've got a glutamic acid over here, right? And it ionizes and it becomes negatively charged because it's acidic side chain. And then that stabilizes by forming an ionic bond with glutamine over here. I don't know if this is actually, this is an example of an interaction where they would be attracted to each other, right? Where you've got a positive charge and a negative charge, and they're gonna attach to each other, and that's going to stabilize that structure, right? You could also have the reverse, or you could have a different acidic and basic amino acid in there, or you could have polar amino acids in there rather than acidic and basic, and it would do the same thing. And we know that this is the case because we have examples of this where you've got different versions of the same protein that instead of having the same sequence in the critical structural feature, they have amino acids that have the same properties in that region, but the sequence has 0% identity, but it accomplishes the same job, right?
Yes.
That means the probability that you're throwing out there is not, I mean, not to put too fine a point on it, the probability that like Michael B here, somebody be like, oh, there's a 1 in blah, 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 blah chance of this happening. That's wrong. Or Douglas Axe is really famous for this because that assumes that there's one sequence and one sequence only that will do that job. And that's just wrong.
Well, the counter to that is there are an infinite number of ways to make cars. It doesn't make them highly probable. And that this is exactly, and so in our Oxford paper, we talked about covariation. So when you change a residue here, a corresponding one is often found elsewhere. And we find those correlations. It's not just our paper, but the bigger community, which is the direct coupling analysis community, leverages that to be able to build their protein folding analysis just by looking at multiple sequence alignments, which is spectacular.
But here's the thing.
AlphaFold, by the way.
Here's the thing. That means that as you have more mutations, you increase the likelihood of finding a stable conformation. More mutations make your job easier, not harder, which is the opposite of what creationists usually argue, that as mutations occur, it can only break things.
Well, I have to disagree because when when we actually have something half functional and you throw more mutations at it, it could break actually what's already partially might have
That
been on the
flies in the
way.
face of what we see in real life, though. Like antibiotic resistance is the reason you can't cycle, you can't do antibiotic cycling to get rid of resistance is because of a phenomenon called compensatory mutations, where you have a resistance allele, right? A resistance mutation occurs, and it has a cost associated with it. You're resistant to the antibiotic, but it has a cost. But then subsequently, you have a compensatory mutation that maintains the resistance, but it recovers the cost. So now, even if you remove the antibiotic from that bacterial population, you don't have selection back to the original genotype, right? Like that's exactly what we see is where you have these two-step mutations happening all the time.
Well, we have microbiologists like Scott Minnick, so he's obviously aware of stuff like that.
Mm-hmm.
It's not applicable to building a complex protein. And we saw that in Lenski's experiment. It was really embarrassing. You had all these things that are DNA repair mechanisms, they're gone. The more it mutated, these cell lines, some of them just started to spontaneously just like die.
That's not true. That's wrong. Nope, that's wrong. You're not accurately citing the paper. So I know the paper you're talking about, the one where it was specifically it was the CIT+ one that later became a mutator phenotype and showed that this was a whole big paper where they said, oh, the mortality rate among those bacteria is significantly higher than the original genotype. And that is true. The mortality in those bacteria was higher. But the problem is there's two things that creationists never talk about when they talk about that paper. One is that the mortality was higher because the population density was higher. And therefore, the density independent mortality factors were the same. The density dependent mortality factors increase with density. Therefore, in the denser citrate metabolizing populations, the mortality rate is higher. That's just basic ecology. That doesn't mean that population was less fit. It was growing faster and to a higher density. The second thing is the populations that died off, the populations that actually went extinct, that creationists love to cite. Those were populations that were grown exclusively on citrate without the glucose, which was the normal food source.
Okay, so thank you for pointing out my mistake. Again, I'm grateful. That's why these conversations are useful. However, so we look at the DCUS frameshift mutation. That's the one that Lenski overlooked, and that's why Minnick was able to be.
That was the earlier one that ended up upregulating some other thing, right?
I don't expect. So you just said more mutations would help it be able to make a functioning protein. I don't expect that DCUS with that 5 base pair deletion is ever going to recover.
Why would it in that environment?
Huh?
Why would it recover? There'd be no selection for it.
Exactly. So you can't make a blanket statement that more mutations is going to increase the probability. We also have all the DNA.
I mean, that's just math, though, Sal. More mutation. If you're trying to calculate the probability of a certain genotype appearing, more mutations per unit time is going to increase the likelihood you find that genotype, right? If you're sampling more of the sequence space in the same amount of time, you're more likely to find a certain sequence. That doesn't mean it's likely. It just means it's more likely.
The question is it going to arrive at that in universal time or geological time? And the answer, I mean, we're looking at it right there. There are a list of about at least 10 genes. One of them, by the way, being topoisomerase, one of the forms of topoisomerase knocked out. Is there any chance of recovery? See, this is what bothers me is you have something that's already a functional gene. It has a few things knocked out. You throw more mutations at it, it's not going to recover. You're actually going to scramble. So this is like having a functional sentence that has a few things missing, and you're going to throw more mutations. It's not going to converge toward something either to that original function, what it's already close to. So that DCUS gene, after 80,000 generations, as far as anyone knows, it's not any closer to becoming a recovered DCUS gene. So
that's fine, because you're
going to end up having proteins, that maybe in one context they might be functional, but it's not going to be functional in that context. And that actually led to this.
I
wanted
appreciate what you just said. That was exactly what I was going to say in that context, because you're talking about the specific context of the Lenski experiment, which in other fora, creationists are very clear that this doesn't represent real life. That's not like real world. That's a fake, lab grown population that doesn't represent the real world, and those bacteria wouldn't survive in the real world. Yes, exactly. So the things that you select for in that environment are specific to that context and are not representative of what you'd expect to see in a more variable environment, an environment with more species, an environment with more organic molecules, because one of the things those bacteria get rid of, right? We see genome reduction, and one of the things they get rid of is metabolic genes for carbon sources other than glucose and citrate, right? That's one of the things those bacteria jettison, because they're never exposed to organic carbon other than glucose and citrate. Now, in the natural world, what happens if an individual cell in a population loses the genes to use some other form of carbon? That bacteria dies because it can't compete with its neighbors who can still use that food source that's available. So there's things we can pull out of the Lenski experiment that are useful. One of them is that, hey, you can evolve irreducible traits. Minnick showed that. But there are other things that you can't generalize because we're talking about that highly specific population. And the genome reduction and the nature of the genome reduction in terms of getting rid of like metabolic genes or getting rid of their DNA repair stuff, the reason you can't generalize that is because you can take those bacteria, as creationists will say in other contexts, they'll say, yeah, you put those in the real world, they die. It's not actually an increase in fitness. Yes, because fitness is context specific and the selective pressures are going to be different. So I don't think we're not disagreeing on what happens and why.
Right.
The implications.
This is where to be a little bit more circumspect, I'll say that the problem is it'd be more credible if we could say this or that will happen in the wild. So what we see experimentally, it's like, can we say that, hey, it's going to be difficult once you have a gene that's disabled, if there's not horizontal gene transfer, that it's going to be able to recover just by throwing more mutations at it. So by way of where I was headed with that is, if you're only 5 base pairs away from making a functional gene, throwing more mutations at this where you keep adding things, you're making the situation worse. Why would you expect it to ever become anything complex?
If it's a deletion.
I'm just saying, from my perspective, from the perspective of people that have jump sides. I was a former evolutionist, not I wasn't like Carissa or Richard Sternberg. I was just a high school accepting it. This is really hard to swallow, that you could build something this complex. And it's like, because I study proteins, and this is like, okay, I'll grant that you could have some of this, like what you're describing with in the microbiology and antibiotic resistance and increase. This is a different animal with a level of complexity. You can't throw those statistics.
So let's
at this level. I mean,
I hear you.
This happens in people that actually study a specific protein, and that's what I'm saying. They're like, I talked to someone who does insulin receptors. He just shakes his head at what he says he sees evolutionary biologists say. And then someone that studies this protein, the specialists are like, their jaw kind of drops and say, well, this is a protein I've studied a long time. It's just difficult. And that's all I can convey is, I said, look, you can't extrapolate the examples you were given to somehow solve this. This again is like trying to say that you could solve a 100-character password because you're able to demonstrate you could solve one that's 2. The degree of complexity counts for something.
Yes. And here's the thing, and we've talked about this before, and last time we spoke, this was actually one of our kind of sticking points, which was, okay, quantify that threshold for us, right? Yeah. If we're saying this thing can evolve, this thing cannot, we need to quantify that threshold, and we need a reason why that threshold is prohibitive, right? Have we made any progress on that front?
Yes. First off,
what's the threshold?
that was the wrong way to frame the problem, because when we talk about theories as far as being probably true and probably not, we don't necessarily have to even invoke thresholds. When we start saying that, this is 90% certain that this is not going to evolve, we don't have to say it's a
You
cutoff.
can't make a claim without some empirical basis, though.
Sure, we can. Sure, no. Well, look,
Mm-mm.
what's the probability that I can evolve a topoisomerase from nothing? I can make that claim. I mean, we know the complexity of the basic parts. There, it's 1,500 amino acids,
Mm-hmm.
and we can, a good ballpark figure is say about 150 of those are probably really necessary.
Mm-hmm. Okay. So
And
do you think it's reasonable?
I'd wager that this is going to be really difficult to evolve.
You
The
wager.
converse of that, from my perspective, if evolutionists are saying this evolved and it's no problem, that really bothers me. Okay.
Do you
If they have
think
the attitude that this is true and you're claiming it's a good science, you're holding yourself to one standard, and you're holding us to one standard, which the community of evolutionary biologists don't even follow
Oh, that's
themselves.
not true at all. Do you think that use whatever example you want, a topoisomerase, a polymerase, which is a big multimeric exam, and ATP synthase is a good example because that's something that's just shared across all domains of cells, right? Do you think that the version of these things that exists now would have been the first one? Like, and I'm not asking you, Sal, intelligent design guy. I'm asking, like, do you think that's what I think? Right? Because
No.
the claim that you are arguing against is that if you were to pluck out an existing topoisomerase, this thing couldn't evolve in the absence of a topoisomerase. What's a topoisomerase? It's an enzyme that grabs onto, cuts DNA, right? That's what a topoisomerase does, right? The topoisomerases that our cells use right now.
Simple. I'm sorry, I have to object because obviously I study topoisomerase. It senses the topology, it cuts, it untangles, then it ligates.
Yeah, okay, so it does, yeah. So
it cuts and it cuts and reattaches.
Cuts and reattaches.
Because
now, I'm not a biochemist. Now, I'm not a biochemist, but correct me if I'm wrong. Most topoisomerases have more than one polypeptide, and the cutting and the ligating are done by different polypeptides. Is that correct?
In humans, it's a homodimer, so you could say two.
Okay, so it's two bits. Okay.
No, this is a good conversation. The cutting and ligating is done by topoisomerase in humans or eukaryotic or most eukaryotic organisms. It is done by the homodimeric complex. Okay. In bacteria, it's a heterotetramer.
Okay, it's a heterodimer. Okay.
It's heterotetramer. It's a heterotetramer.
Even better. So here's the thing. Do you think that I think the first word, because going back to talking about de novo proteins, right? De novo protein families, right? Do you think that I think the common ancestor of all those topoisomerases was a heterotetramer?
No, I don't think that.
No, of course not. It would have been a single polypeptide. Because if you look at obviously it's a homodimer, right? Homo means for everyone watching, homo means it's the same, right? So those are homologous genes, right? They share a common ancestor. There was a duplication, and now you've got two subunits, right?
I'm sorry. It's not completely fair to me to I'm not trying to direct this criticism at you. You're asking me to get in your head and say what you think.
That's fair. So, right. So
fair. I mean, I understand why you posed it to me. And you're going to get me tripped up if I try to mind.
That's fair. Yeah. So let me just make my point straightforward then. My point is, I'm not saying out of nowhere you're getting this protein as it exists now, right? You're getting an ancestral state of that protein that is less complex, that's less specific in its activity, but it still has the underlying activity, right? Okay. That's the idea. So calculating, there's two problems here. One, calculating the probability is a problem. You need to have an empirical basis for calculating that probability. But two, calculating the probability of the extant version of that enzyme does not represent the probability of it originating, because its original form would have been less complex. And we know that this is the case. We can see this when you have new functions evolving in enzymes, in proteins, not necessarily enzymes. You often see a less specific form of that activity adapt into a more specific form, right? And we have examples of that in protein evolution.
So I don't quite know how to frame this, and it's something I've been working on. And it was great when I was on your show. We talked briefly about nuclear localization signals, and you said that's a good paper. You should write that. I've actually explored that. I've had some nice discoveries. This relates to the question, can we take the extant and use that as a model for what the original is? Or try to make a hypothetical ancestral protein.
Mm-hmm.
And one thing I didn't get to in the debate, I said, you have these nested hierarchies where you have the phylogenetic trees where they
Mm-hmm.
modify. I said, when you have an unconstrained mutations where it's modifying what we call the neutral regions, the unconserved regions. Okay, I don't know.
Yeah. Unconstrained is a good word. Yeah.
It becomes a different issue if it is constrained.
Right.
One of the things I looked into after I was on your show, I said, Dr. Dan did say he was curious. I said, I respect that. He thought that was a good question. And I think he wants to hear the case that I could make. I started to look at the important proteins because that is the one that governs what the nuclear localization signals will be throughout the entire cell or creature. And this is something that actually bothered Richard Sternberg. Richard Sternberg brought that up. That's why, he alluded to it in some of his work on signs between mice and rats. And someone else who will not be mentioned was also pointing out all the binding proteins, the CTCF binding proteins have to be modified. And I know this is kind of very long-winded, and I apologize for that, but this is really the most complex thing, is that what changed Tan, I showed her picture there. She was an evolutionist from communist China, creationist, and she was studying molecular biology. She was looking at, you mentioned polymerases. She said there's not one that, I said, are they conserved? And I was shocked. She said, sequence-wise, no. There's no, you'll find these exceptions. So like you have, and you mentioned this, like, do you have to have the same sequence? And I say no, and you say no, so we're in agreement. So one good example is serine proteases. Serine proteases have the same tertiary structure, same shape.
Right.
They have totally different sequences. That is where you can get convergence.
And that makes them easier or harder to evolve?
Superficially easier.
Easier. No, not superficial. Mathematically easier because there's more.
But if this is where it goes into constraints, it's going to be very difficult to evolve. I don't know specifically about serine proteases, but if we're looking, like, let's say, at the importin, and it's going to be governing all the localization signals in the cell or any transportation process, you modify that. So you build a phylogenetic tree of all the important proteins out there. And you say, hey, okay, what are the binding properties here? If you modify that and you have phylogenetic trees for all the proteins and their localization signals, they have to co-evolve. Co-evolution. Yeah. And I'm just sharing this with you. I've shared the problem of co-evolution with some of my colleagues. And when they saw it in evolutionary literature saying this co-evolved, we all just laughed. Because the probability of changing and I'm just pointing out
how many
mindset. We saw that if you change the policy, the localization signal,
right?
Because you're going to have to simultaneously change it for all of these. And so we were talking about spliceosomes going from like a yeast spliceosome to human.
Well, Sal, can I jump in before we get to another specific biochemical example? Because I have a question for you that's independent of the specific examples. And you're telling about, okay, your colleagues are talking about this. You see that like these mutations would be dependent on each other. They'd have to co-evolve or it wouldn't work.
That's where the probability. That's
my question is this.
If I could just complete one thought.
Yeah, go ahead.
The reason I'm putting that on the table, this is where I felt the probability argument was more pointed. So like you said with the serine proteases, I said it's easier, and I'm conceding that if it's unconstrained. So I had to look at constrained situations, and that kind of, for me, that kind of made the argument more rigorous. Because just like a car, there are an infinite number of ways of making a car. But what makes it improbable is the parts have to fit together and connect. And this is where the probability argument I felt was a little bit more rigorous and a little bit more pointed.
So a small point I want to make is, okay, I see where you're moving towards having like some kind of standard here, but there's still not, at least correct me if I'm wrong, there's still not a study that's been reviewed, people checking the math of like, here is the standard, here is the threshold, here are the probabilities, right? That does not yet exist. That should exist. Somebody should write that.
Totally agreed.
Okay, that's a small point.
That's on
small
me.
Point.
And you had total right to call me out on that, and I have no problem with that.
Small point. That bigger point that I want to make here, and then I think I'm going to jet after I make this point, and then I hear from your response to it, and then it's late. I'm going to go to bed. So you're having these conversations with your colleagues. You're obviously citing papers, right? You're saying, hey, look, someone did the structural studies. They found that this amino acid is dependent on this one. If you change this one, you have to change, right? You're looking at data saying, here are the dependencies in these structures that constrains what mutations are compatible with what other mutations, right? You're looking at this stuff.
And if I may just say,
yeah, go ahead.
That actually ties into our published papers. We're actually building these algorithms to detect covariation.
Cool.
This is actually something central to professional work. Okay, so I'm just letting that's not for you as much as the audience. Right. This is something we're actively studying. Okay.
Right. So you're saying, okay, look, there's covariance here. And then you all kind of say, oh, that's interesting and important. It has implications for the evolution of these systems. Are you talking in these conversations to evolutionary biologists who are working on these things? Like, are you reaching out to the authors of these papers? Are you reaching out to people who work on protein evolution?
I'm sorry.
You should do that.
Okay, I think
you all should do that. Okay. Because this goes back to kind of what I started with like creationists not like being correct about the things they're talking about, which is because like you all got to get out of the room where it's just you all talking to each other.
Okay, so
this doesn't count. Like, I mean, professionally.
Okay.
Like, go to the real conferences. Go to the evolution conference every year.
This is hard. This is a hard conversation to have. We ought to have. I acknowledge you and I ought to have. Other people ought to have at a more professional level the conversation that we're having here. This is fair. We're interacting. We're kind of thrashing it out and criticizing each other's ideas fairly.
Give a talk at a conference. People would accept it. Do a poster.
Okay, so I'll tell you this. The situation is we just despise some of them.
Too bad. Science. Have you met scientists? We hate each other. People get in fistfights over nonsense at conferences.
So I think
that's
all right.
That's not an excuse.
I'm not saying that you're wrong. I'm saying you're right. We should talk. But there is definitely a cultural divide where we despise some of these guys. We laugh at them.
That's too bad.
But you're saying we should talk, and I have to say, well, Dr. Dan's right. The other thing is, especially now that I'm trying to write good papers and textbooks, I should write and communicate to these guys.
Yeah, you should. Okay. Yes, if you're saying this paper has this implications, you should reach out to those authors and see what they think.
Okay.
You should do that because look, Cassell, you and your team and whatever lab you're working with, whatever people you're working with, like you want to be either in actuality or in perception, right? Participating in the broader endeavor of science, right? And the thing that you're doing is you're going to do science and you're going to show that these specific set of ideas are wrong and these specific set of ideas are right. If you never bring those ideas to an evolution conference, if you never put them in a real journal, nobody is ever going to hear about them. Nobody is going to care. And critically, nobody is going to evaluate them.
So if I may say this, Dr. Dan, part of that hostility on our part is we've had papers where the editors told us we're right, but we're not going to publish. Okay, so
go to a conference. Conferences,
okay.
Go to a conference. That's like one step. Do a poster at a conference. They'll accept. I mean, I have seen some of the biggest nonsense on posters. in my life. Okay. Like you do a poster saying XYZ can't evolve because of these probabilities. Like that will get you in the door. And then you spend the poster session, you're talking to people and you're making your case, right? And then the next year you give a talk and then you put a couple papers out for peer review. I'm sure that you could, I mean, look, some of the stuff that's in like the Journal of Theoretical Biology, the journal, like, come on, like you could get this stuff out there and like, you should be trying to do it. And like the creationists need to like, you know, gird yourself to like deal with reviewer number 2 because that dude is always a jerk. Doesn't matter who it is. Reviewer number 2, always a jerk, right? And just like do it. If you think you're right, creation is very confident. You're sure you're right, right? So right, you don't even have to read the paper sometimes. You know you're right. You don't have to read the math because you know you're right. Put it out there. Okay. And convince people because it, you know, we do change our minds. We do change our minds about stuff.
Well, okay, so not that I'm saying you should love the guys, but Sternberg, Beckley, they're probably lesser known names. And not
yeah, but like, see, but here's the thing, like Beckley's a jerk to people. Like if you read Beckley's blog on Discovery Institute, like he's like the Professor Dave of creationists over there. Like he's a jerk to people. Why would anyone take him seriously? He's not putting papers in evolution journals. He's not doing talks at real conferences to try to convince people. He's name calling on his blog.
I do have to say something though. Biology journals, we think, you know, we want to put it, there is, I will just say this, the venue that is more appropriate would be journals in computational biology, protein biology, statistics, mathematics.
And the evolutionary biologists aren't going to see it, and you're never going to make your case to the people who you're trying to convince.
We've given up trying to convince them.
Then you're not doing science. So then you're not doing science, man.
Well, see, there are all these biomedical researchers that are moving forward, like Robert Matheny. He's doing a lot of great stuff with being able to regrow heart parts, and cardiac thoracic surgery. And
yeah, cool.
Just
Discovery Institute.
There's more to science than just dealing with evolutionary biologists.
Yeah, but Discovery Institute said back in the mid-90s they were going to overturn the dominant paradigm within 20 years. The 20-year mark for that was 10 years ago. How are they doing? Have they overturned the dominant paradigm? It hasn't happened. Why not? Because they publish in their in-house biocomplexity journal instead of trying to get their papers into real journals. They're not trying to convince anybody.
Yeah, I thought Stuart Burgess did well publishing Institute of Physics. He took down Nathan Lentz pretty hard, and I wouldn't expect him to publish in an evolutionary biology journal, but he took it to the right people in biomimicry and bio-inspired design. And I felt he made the case quite convincing. And that's probably more the route that I would probably go, because then we're actually dealing with our peers who are actually knowledgeable about physics and mechanics.
But Sal, that attitude is the problem. Okay. You've got to talk to people who don't already see things the way you see them.
I think that's fair, Dr. Dan.
Right? Like you got to make your case to the people who you're saying, you know, there are gatekeepers who are keeping you out. Those are the people you have to convince. Right? And when I say you, I don't mean you specifically. Yeah. I mean like creationists as a whole. You know, Jeanson will say this, Carter will say this. Creationists as a whole will say, well, we can't get heard. You said a few minutes ago the editors won't even look at your papers. Those are the people.
They looked at it. They said our conclusions were right, but they're not going to publish.
Okay.
So it was very disturbing, and it had to deal. I've talked about this. I need to just relate something in my own little universe. For 7 years, we tried to push this paper critical of nylon eating bacteria in its evolution, and the editor said, "You're right, but that's old hat." And then we see Carvunas published in 2021 or 2019, citing a paper that we'd refuted, but the editors wouldn't publish. It gave me a very bad taste. I said, look, they're not self-correcting here. This is a big deal. for us personally, and we worked hard on it. And we had, I said, do bad ideas just keep having such a free pass? That would be intolerable in an engineering context. If you describe something that's going to work and it doesn't, that's it. It's game over. But I'm just relating to you why we have some of these frustrations. But to what you said, should I go to an evolution conference? I think
Yeah.
you're right. I should.
Get out there, and not just you, and not you specifically. I mean, like creationists, like Nathaniel Jeanson, like hasn't done anything since he left grad school. Like he's written a couple books and published for AIG's blog, right? Like
Yeah.
he wants to overturn the paradigm. Well, like put in the work, bud. You know how it works. Michael Behe's out here writing books every couple of years. He hasn't published anything in ID since 2004. That one paper, right? It's been 20 years, and like, you know, it's a lot of nerve for all these people to say, oh, we're not being heard. Nobody's listening to us. My guys, you're not even trying. You're not even trying to have the conversations. And like part of that is even just within your own conversations. Like if you're talking about something, email the people who wrote it. Hey, here's what we think the implications of your paper are. What do you think? You think this is fair? And see what they say. And like,
You know,
creationists don't do that. And when the authors do, the creationists will often reject what the authors say and say, well, that's just your opinion. I have a different opinion of your own work, right?
Well, actually, as much as it pains me to say it, I think you're right that we or collectively.
You know, collectively, I don't want to put this on you. I mean, to your credit, Sal, look, here, I'm going to give you credit because like most creationists don't even have these kind of these informal conversations, right? Like it's to your credit that you do, but like even, you know, that's more than most people do. The vast majority aren't doing any of this. They're just kind of in their little bubble, right? And like, you got to get out if you want to convince people. If you're doing real science, you got to get out and actually do it.
Well, I'll just tell you this. See,
and then I'm just going to say what you're going to say, and then I'm going to say goodnight because, man, it's late.
Well, first off, I'm deeply appreciative you came here. Unlike other creationists, I'm not eager to save face in one conversation. They're always so eager to defend their position, never admit they could improve in some place. That is not me. And I will try to maintain that, you know. It gets me in trouble with some creationists that say you give away too much. It's like, well, look, if the other guy is right or could be right, I have to consider it. And so I'm just speaking to the audience here. It doesn't set a good example if you always act like you know it all and that you can't possibly that you're infallible. That's not how things move forward. And so I think a lot of my material has been improved by discussions like what we've had here. You'll say, look,
that's how science works. Everybody should do that.
The argument, as stated, wasn't the argument you're really making. I said, okay, I could reword it and say this is a different argument. And that's, you know, and I think, honestly, I think some of my stuff is going to be the best creationist stuff out there because it's gone through this vetting process, at least better than.
And if it goes through peer review, that will be an even better vetting process. If you deal with people at a conference, that will be an even better vetting process. I am like, you know, some rando biologist. I'm not like a big name in the field, right? You go to a conference, though, like I told you, I was at that talk with Grauer. You go to a conference, you can chat with these people. You can tell Grauer to his face. Like, you know, like, come on.
Yeah. So, I don't have, okay, just from a logistic standpoint, just so you know, that's not in the cards immediately because being in a PhD program is.
I mean, this is turning around to, you know, an aircraft carrier, right? Like this is a big, not just for you specifically, but for the field as a whole, right? This would be a change in approach that would take significant.
If we could just end on a nicer note. I mean, this is kind of painful for me.
I'm sorry. I didn't mean you. I was talking, you know, you're just the person who invited, you know, and to your credit, you know, you have these conversations, so then you get to hear all the stuff that people fling in that direction.
I had a burning wanting to talk about, okay, before we say goodbye, I just wanted to talk about cars a little bit.
Just for a couple minutes, and I got.
We were having so much fun before. Yeah. The last live show we did, that we actually started late because we were talking about the vacuum cleaner car at the Can-Am races.
Yeah, and I got my Group Bs up there. I got all the Group Bs up there. my Group B rally cars, which were bananas because they was basically unregulated. Yeah, it's
I
serious
love
as
that
fly.
vacuum cleaner car. And so I thought of,
The
I
vacuum,
wanted
that
to
was
tell
cool.
you, the drivers are passing out.
Yeah, from the G's, right? They're blacking out in the corners. Yep.
And I'm just like, okay, I've been in an airplane. I pulled 6 Gs. I started to black out, but I could still control the airplane. For a driver to be pulling that many Gs in a turn, it's just insane.
That's
I mean, I'm
wild.
surprised the car just didn't go off the road.
'Cause it's-
So it was dangerous in the fact it was such a good car that it basically caused the driver to black out. And just like, you're talking, you're talking 8 or 9 Gs. That's not easy to do in a car.
No, you got to be
And
moving.
even in an airplane, I could take 4 Gs, and that when I drive on the road, if I just start to slip a little bit, like say even like fractional Gs, I'm just like, I'm going too fast.
Yeah. Oh, yeah. Totally. Yeah. You shouldn't be doing that on the road, right? That's the slow down signal, right? That's
Yeah.
Newton telling you to take it easy.
Yeah. So okay, that was it. I can't. So we've been at it for an hour and
I
39
know. We've been
minutes.
talking a bunch. Yeah.
I cannot thank you enough for the time you've honored the channel by coming here. And I think this is one of the best conversations I've had in a long time.
Yes. Thank you. Thank you for having me on. I really appreciate it. I hope it's clear that I'm not coming at you personally. I hope that is clear because that is not my intention.
Oh, it's very clear.
Okay. Cool.
And maybe someday we'll have some other conversations.
Yeah.
I would look forward to it. And I have some work to do on the things you suggested, and just wanted to express my appreciation. And thank you, Dr. Dan.
Have a good night.
You too. So, guys, Dr. Dan has to turn in, and I'll probably be turning in too. Let me just say hi to all the people here. We have C. Brown. And can we talk about theoretical speeds the Earth is allegedly moving? The Earth is moving at about 300 kilometers per second relative to the galaxy. There are experiments. I really do think, because I showed briefly on my, see if I have it. I worked on special relativity, and I wanted to show it. Yeah, here it is. Hang on, I'm gonna screen share. Let me get my screen share going. One moment, let me get my screen share going. I want to address this question. Let's see. And so that was an interesting question. And then I have to. So I was talking about electromagnetic theory here. Classical electrodynamics. And you can take that. It's a painful derivation. It took me 8 weeks to formulate this. You can show special relativity here. With special relativity, with some concepts of general relativity, I think you can estimate the absolute velocity of the Earth. I have to check with Philip Dennis, but you can actually, depending on what this equation was really important is the faster you move, your clocks change. We know that. We have to adjust the GPS satellites. And there are a variety of other experiments. We can probably establish then about where a zero velocity, we can make a zero velocity reference based on clock speeds. If we have cesium beam atomic clocks, and then we can estimate the actual velocity of the Earth relative to absolute zero velocity. Some people will say, no, that's impossible. I'm going to dispute that because when you accelerate, you move into a different inertial reference frame and it affects your clock. There is a point when if you start accelerating in the other direction, you reach zero, and then and if you keep going, if you keep accelerating, your clock will start to slow down again. So if you're moving fast and you try to, decelerate, your clock will speed up. But as you keep going the other direction, if you cut your rocket engines the other way, you're going to start making your clock move slower again. And there are ways we could do this. We kind of building the instruments to do this would be hard, but I think we could do theoretical work on that. So electromagnetics are not gravity. That's a theft of identity. There is still a relationship between special relativity and electromagnetics, and that was I'm not making that claim in a vacuum, because this was actually something that was assigned as homework to all the grad students in my class. This derivation here, it connects electromagnetic theory to special relativity. Special relativity deals with inertial reference frames with no acceleration. Acceleration goes into gravity. When you have the phenomenon of acceleration, it goes into gravity, and that's the general theory of relativity. So that's just a different, I don't know. I was trying to see if that could be unified with gravity, and that's something way over my pay grade. But we do know special relativity can be demonstrated through study of Maxwell's equations. Whether that can be extended to questions of acceleration and gravity, I don't know. So let's see. So, Sal, do you think Dan Grauer is being pressured in a sense to say that his math is incorrect? And do you think the 2020 paper was a rescue? Okay, I have my opinions, and I promised Dr. Dan I'd read Dan Grauer's stuff. I have an extremely low opinion of him, and I'll tell you why. He went on a rampage against the ENCODE project, and the ENCODE project were people just trying to solve heritable problems with heritable diseases, solve cures for cancer. They're just doing their job, exploring the epigenome, and he's calling crooks and ignoramuses and just really nasty things. I said, that's so unbecoming. And I said, let the guys just do their job. These were guys from prestigious institutions, from Harvard to Johns Hopkins to Stanford. And that's why I was saying I had just very little respect for him, because he was criticizing other scientists who weren't necessarily creationists. Probably most of them were evolutionists, and they, imagine their shock. So the question is, do you think to say that his math is incorrect? And all I could say is, I don't know. I will tell you this. I think my math is correct, not because I'm a genius, but this is stuff that has all the way since the 1950s. The math has been there since Hermann Muller wrote Our Load of Mutations, and Hermann Muller won a Nobel Prize. I don't see how I could be wrong. So I promised Dr. Dan I'd review the stuff he's been sending me, and I'm going to look at what Grauer had to say, where they are correcting his math. I will say this. I did see that his numbers were a little bit off from the ones that I came up independently. And I said, okay, maybe that's just a rounding error, but it still looked kind of wrong. But I said, I went through the use of the Poisson distribution as well as I could. So we're going to look at this. And Mark O'Connell, Tangela, it's great to see you. Thank you, Vesper, for visiting. You got to see a creationist talk to an evolutionary biologist. So I know this isn't a nice thing to say, but Max was just a buffoon compared to Dr. Dan. Dr. Dan is brilliant. I consider him a peer. He's a biologist. I respect him. We disagree pointedly, but at least his stuff is well reasoned. Max, who I debated, I'm just like, dude, you don't even have basic molecular biology. You're just not. You're arguing. it's you're arguing about stuff you don't know. You know. And thank you for the kind words here. And Otangelo, great to see you. And so, let's see. And Damian Martinez, great to see you. Thank you so much for visiting. I don't do this very often. Anyone else loving the ships over creation myths left shoulder? I didn't even see him, so thank you for pointing that out. Yes, I think. Yes, Dan is helping me, and I want to hear what he has to say. Yeah, yes, we are all very grateful. So, Nestle, always great to see you. I'm hoping I don't miss anyone. I saw some old friends that I haven't seen in a while. At least want to acknowledge them. I'm scrolling through that right now. I think I saw Andrew Kaufman earlier. Yes, where are you? Did I just miss you? I feel bad. There he is, Andrew Kaufman. And I don't see anyone new or an old friend. I think we've gotten through most of the list. Here's one. And Ruth was here. So great to see you. Ancient. Oh, well, thank you for visiting. Thank you for the kind words, Ray. And Unfinished Journey. And I tossed Max. He's just a troll, in my opinion. I have no use for him, except he helped make the debate possible. Sorry to be so rude, but I just have not a lot of respect for his conduct in the debate and his attitude. So anyway, guys, it was great. I'm gonna just